Assalamualaikum, my name is Aiman Firzana. We are from Group 10 and we are going to present our topic, which is cell-based immunotherapies, or CBI for short. CBI are revolutionizing how we tackle serious diseases like cancer and autoimmune disorder. Take CAR-T therapy for example. It's a groundbreaking approach where T cells are extracted from a patient, genetically modified to target and destroy cancer cells, particularly effective against blood cancer like leukemia. Then there's dendritic cell therapy, which train immune cells to recognize and attack cancer by capturing cancer antigens and presenting them to T cells. Although these therapies are highly promising, challenges like side effects, high cost, and complexity remain. However, ongoing research continue to address these issues, aiming to improve safety and expand their applications. This progress highlights how CBI is revolutionizing medicines with personalized and effective solutions. Let's dive deeper into how these therapies work and their broader application. That's all for me. Now I will pass it to Danish.
Having researched cell-based immunotherapy, there are 4 approaches in this case study, which is the first one is the Chimeric Antigen Receptor. This method is used to treat leukemia and lymphoma. The next one is dendritic cell therapy. This treatment is used to treat including melanoma, prostate cancer, and ovarian cancer. And the third one is Adoptive Cell Transfer with engineered T-cell receptor, ACT. This method is used to treat cancer like melanoma and lung cancer. And the last one is tumor-infiltrating lymphocyte TIL therapy, which used in treating metastatic melanoma and several other cases of cancer. Now, for the properties of the material, for CAR T-cell therapy consists of viral vectors, culture media, and cryopreservation solution. Viral vectors in this method consists of 2 different kinds of virus, which is the first one is retrovirus, and the second one is lentivirus. Next, the second is dendritic cell therapy, consists of cytokines and growth factors, antigens, and adjuvants. The third one is Adoptive Cell Transfer ACT with engineered T-cell receptors. It's of viral vectors, which is the same as CAR T-cell therapy, which is retrovirus and lentivirus, culture media, and the last one, cryopreservation solution. And the last one, tumor-infiltrating lymphocyte therapy, which is consists of cytokines and growth factors, antigens, and the last one, adjuvants.
Recent development, we have advances in NK cell-based immunotherapy, where NK cells are equipped with TCRs to target tumor antigen precisely. And then we also have the enhanced FC receptor to improve NK cell ability to destroy antibody-coated tumor cells. Then we're going to talk about the model and mechanism of CRT cells. Engineered CRT cell recognize tumor cell through CARs binding to TAA. And then after the recognition, CRT cell target tumor cell by releasing granzymes, perforins, inducing trails, and VSL expression. After that, the immune cell penetrate the tumor microenvironment by stimulating pro-inflammatory cytokines and chemokines. After that, we're going to talk about the model and mechanism of CAR NK cells. CAR NK cell express Chimeric Antigen Receptor to identify TAA on tumor cells, and then they target the antigen like CD90 for blood cancer and EGFR for solid tumor. Upon binding, CAR NK cell release granzyme, preferins, FSL, and trail to induce tumor apoptosis, and then CAR cell is gonna eliminate suppressive cell like MDSC. And next, we have the advancement of the cell-based immunotherapy. The advances include technological innovation, where advances like CAS9 enable precise immune cell modification. And then we have the combination of therapies, which combining immune checkpoint inhibitors with virotherapy and anti-angiogenic. And then lastly, we have the CAR NK therapy for autoimmune disease, where the CAR NK cell is engineered to target autoreactive cells, sparing the healthy issues and reducing side effects compared to the traditional therapies.
Thank you, Haddad. My name is Priya. Now I'm going to present the first part of the discussion, which is application of CBI. We all can tell that CBI is applied in the cancer treatment to have special antibodies and target and destroy the melanoma cells by suppressing their growth and blocking the checkpoint protein. In autoimmune disease, TARM1, T-cell interacting activating receptor on myeloid cells 1 binds with the type 2 collagen to suppress erythroid arteritis via dendritic cell maturation. For infectious diseases like COVID-19 and tuberculosis, engineered macrophages are produced to treat the critical pathogens using a bioreactor. Next, as for the limitation of CBI, patient tends to develop side effects such as cough, dizziness, rash, as the immune system reacts with the cancer. Moreover, CBI treatment is quite expensive due to the sources required to manufacture the CAR T cells according to the patient's specific gene. Then sometimes, chronic antigen exposure may lead to the development of exhausted TCD1 cells and self-renewing stem cells. Next, I would pass it to Syafiqah.
Thank you, Priya. My name is Syafiqah Riza. Now I'm going to present the advantages of CBI. Firstly, enhanced tumor targeting. According to a study by Yen Ridley in 2024, genetic engineering of immune cells such as CAR-T and CAR-NK cells enhances their ability to specifically recognize tumor antigens. This precise targeting ensures that cancer cells are effectively eliminated while sparing healthy tissues. Secondly, enhanced cytotoxic potential. Engineered immune cells demonstrate superior potency in killing tumor cells. This modification strengthens their binding to cancer cells, triggering the release of powerful cytotoxic molecules like perfins and granzymes. This mechanism ensures efficient tumor destruction. The third advantage is short treatment time. One of the most patient-friendly features of CAR T-cell therapy is its efficiency. Unlike traditional cancer treatments, requiring months of rigorous chemotherapy, CAR T therapy involves a single treatment session and typically no more than 2 weeks of inpatient care. This can reduce both physical and emotional burdens. Last but not least, the advantages of CBI is personalized and precise. CBI treatments are tailored to individual patient needs. By leveraging genetic profiling and tumor biopsies, clinicians can identify the most relevant targets for each patient. For future indications for cell-based immunotherapies, which focus on the first one, the enhancement of CAR T-cell therapy, the second one, optimization of dendritic cell therapy, the third one, focus on targeting solid tumors, and the last one, focus on improved delivery systems.
Thank you, Syafiqah. Now I will present the conclusion for our topic. In summary, CBI, particularly CAR-T and dendritic cell therapies, represent a revolutionary shift in the treatment of diseases such as cancer and autoimmune disorder. By harnessing and enhancing the body's immune cell, this therapy provides a more personalized and targeted treatment approach. While challenging slight side effects and costs remain, ongoing research is driving progress. These advances are making this treatment safer and more effective. As the field continues to evolve, CBI has the potential to become standard care, offering improved outcome and new hope for the patient worldwide. That's all from our group.